Alright, ninth grade biology students. In this video, I'm going to be talking over mitosis and meiosis. So, what is mitosis and meiosis? That's going to be the point of the video. We're going to compare them. We're going to talk about the processes that make up both of them. So the first thing we're going to talk about is where these processes occur. So, as you already know, you guys are smart ninth graders, probably already talked about the cells. So the nucleus is where the genetic information is stored. So there's all your genetic information in there. So what we're going to start off is just defining chromosomes and how they're formed. So in your nucleus of a cell, here's a eukaryotic cell, you got your organelles in here, blah, blah, blah. And here's your nucleus with the genetic information just kind of boiled up, coiled up in there. So if we take a closer look here, here could be a strand. And so we have these just long strands of chromatin, super thin, and it's called chromatin, and that's just the genetic information. And it's long strands, and they're wrapped around these proteins. Kind of wrapped around in there, and these proteins, these little balls that I drew, these are called histones. You don't need to know that, but that's just a quick example of what this is and like how it's going to form later on. So that's that, the chromatin wrapped around the histones. Alright, so now we're going to take a closer look into what a chromosome is. Just define that real quick. So that genetic information inside the nucleus, so first we're talking about mitosis. Alright, so we have chromosomes that are going to form from that genetic coiled up information, and they're going to form these chromosomes. These are super dense, compact, and I'll draw it in the red again so you know that this is just super dense and compact genetic information. That's the chromatin wrapped around the histones. And so that's basically what that is. And so this is a chromosome. So what is a chromosome? Well, it's like I said, I think I spelled that wrong. Something like that. So what is a chromosome? Well, a chromosome is going to be tightly packed genetic information, and it's going to have genes on it. And what are genes? Well, genes are your traits and characteristics by yourself. So we have a chromosome here as an example. And on the chromosome, you're going to have sections broken down. So I have this section here. We can call this section A and B. And so, for example, this section, this is a gene. A gene is just a section of the chromosome. And genes are going to make up traits and characteristics about yourself. So let's say A could be the trait for what color eyes you have, eye color, something like that. Anything about you. It could be how long your nails are, or something, I don't know, how smart you are. It could be intelligence, just traits about you. So that's what a chromosome is. All right, and so the genes are going to be on these where the DNA is coiled up here, and in the middle, this little ball looking thing, this is just kind of the middle of the chromosome, and this is called the centromere. I don't know if you can see that. Centromere. Okay. That's that little ball. That's called the centromere. Alright, and you're going to have to know that later, so that's just kind of like the middle, and this is where the sister chromatids will attach. We'll talk about that in just a second. So that's your centromere. You have your chromosome broken down into genes that have traits. So that's basically what a chromosome is. And these traits, these genes, so these are just called alleles. That's the official term for it. So this would be your allele A for eye color. So we can just write that down. These are alleles. And basically the location on the chromosome is called your gene locus. So it's located, it's located at the bottom of the gene. So you'd say it's gene locus, genes locus or loci. That's its location. So that's where the allele is located. That's where the gene is located. That's going to make up who you are. All right. So now you know what a chromosome is. We're going to go into mitosis first. So what is mitosis? What's the purpose of it? Well, the purpose of it is to, sorry, I kind of spelled that wrong, create more cells. And these are going to be identical cells. So let's say you got a cut on your leg or something and you're bleeding and there's missing skin and it's just super gory. Your body's going to want to replace those so you don't have a huge hole in your body. So what's going to need to happen is your cells need to make more cells, and the way that your body does that is through mitosis. So what is mitosis and how does it happen? Okay, so you have your cell here, and with the nucleus, as I showed you before. So normally your cell is an interphase, and that's just when it's doing its normal functions, and you have your genetic information just coiled up. So that's most of the cell's lifespan is usually in G0, and it can't start mitosis. You don't need to know that, but when your body detects that mitosis needs to happen, it's going to basically tell this cell that mitosis needs to happen, and it needs to make more of itself, basically. So how does this happen? Well, the DNA here is going to turn into those chromosomes. So instead of being tightly coiled up and just super bunched up, it's going to coil itself up into these nice pretty chromosomes. Humans have 46 in somatic cells. And also mitosis only happens, put a little star here, only happens in the somatic cells. So what are somatic cells? Somatic cells are cells in your body that have 46 chromosomes. So humans have 46 chromosomes. And these chromosomes are what I showed you before. The genetic information is on those, and we have 46. And so that's what a somatic cell is. The other one is a gamete, which we'll talk about when we look at mitosis. So here you have your DNA that got bunched into chromosomes, and so to make another one of itself, well, if it just split into two right now, then you would have two cells with 23 chromosomes, and you can't have that because the cell won't work unless it has 46, because humans need 46 to live normally. So what it needs to do is create copies. So here we have 3, just to simplify it. So now it needs to make 6 chromosomes before it can replicate. It needs to duplicate the genes on the chromosomes. So there's still going to be 3 chromosomes. Here, I can draw this out. All right. So we had those 3 chromosomes to start with, and now the cell knows it needs to do mitosis. So what it needs to do is it needs to duplicate these the information on these so that it can be transferred to the other cell. So what it's going to do is replicate this, and here you have your centromeres here. And so what's going to happen is this, the DNA was already replicated before, and so it's going to take those, the extra DNA it has, and it's going to make the, it's going to look like this, kind of bunched up in the middle there. And so now you have 2 of the same alleles. It's kind of looking a little funky here. And so you probably recognize this. So now, you started with 3 chromosomes, and how many do you have now? Well, you still have 3. So each one of these is still one chromosome, it just has double the genetic information on it. And so we call these sister chromatids because they're identical. Sister chromatids. And this is the vehicle in which the mitosis is going to happen through. So they're connected at the centromeres. Alright, so now that your chromosomes are duplicated here with your sister chromatids and they're combined at the centromeres, what's going to happen next is, I'm going to erase this. What's going to happen next is you have your cell here with your nucleus. You've got your chromosomes in here. Not very good at drawing these. With your sister chromatids, I think I had 3 before. We'll draw 3 out again here. Boom, boom. All right. So now that you pretty much have, these are connected here. Sorry. Okay. So now that you pretty much have double your genetic information, the cell can split into 2, so that it has the same amount of DNA from when it started. So what's going to happen is these chromosomes, they're going to line up. They're going to line up right on the polars here. So it's going to be boom, boom, boom. They're going to line up along this dotted line here. And so remember, they're connected at the centromeres. And so you don't need to know this, but the spindle fibers are going to come connect here. Boom, boom, boom. And they're going to get ripped apart. All right. And so now the sister chromatids are going to get ripped apart. So this one's going to be over here, this one's going to be over here, this one over here, this one over here. Boom, boom. Dotted line down the middle. So it got ripped apart. And now this is going to form into 2 nucleuses. So now you're going to have the 2 nucleuses here with your 2 things of chromosomes, 2 chromosomes. I think I had 3 to start with, but now we have, we'll just say we had 4 the entire time. So, so now you've got your chromosomes. So you started with 2 chromosomes, and now it's split. So now you have 2 chromosomes in each nucleus, and then the cell's going to do cytokinesis. It's going to divide, and now you're going to have 2 different cells, but they are going to be identical with the exact same, write this out, same chromosomes. All right, so that's what mitosis is. So you start with 1 somatic cell in your body, and it turns into 2 identical daughter cells. All right, so now these cells can go off, do their job, and then they'll come back around, duplicate their DNA, replicate the chromosomes to have sister chromosomes, and then they'll go through the mitosis process, separate, and that's how your body keeps a good supply of cells in your body when they need them. All right, so now we're going to go over and talk about meiosis, which is more complicated. Meiosis. So it's going to be similar steps in the beginning of mitosis for meiosis. So you start with your cell that has that genetic information coiled up in here. And this is still a somatic cell. Somatic cell. Okay, so this is going to have the 46 chromosomes in humans. And so meiosis, what meiosis is, the purpose of it is to create sex cells. We're going to call these gametes. So mitosis, the purpose over here is going to be to make gametes. And gametes are your sex cells for your body. And these are going to have half the amount of chromosomes. So normally you have 46, and that's where we're going to start with the somatic, and then we're going to go to 23 for the gamete. And that is the purpose of meiosis, along with other things we'll talk about, like genetic variation. Okay, So the first step is the same. It's gonna bunch up that DNA in there, and you're gonna get your chromosomes, and they're gonna be nice and pretty in there, and then it's gonna replicate them. Make this a little bigger. It's gonna replicate them, and you're gonna have that iconic chromosome shape in here, all connected to the centromere. So, for example, we'll say this cell is 2. Humans have 46, so this cell, we're gonna say it has 2 chromosomes, and it just duplicated its DNA, and now it's got the sister chromatids here. Each one of these is a sister chromatid. And you still have two chromosomes. So you started with 2, you still have 2, you just have double the information there with the same information on these. So this side is gonna have the same as that side, like I said before. So next, what's gonna happen, this is where meiosis gets a little bit different. This is where it gets a little complicated. Okay, we're gonna draw this nice and big here. Alright, so I went ahead and drew this out. So this is where we compare meiosis and mitosis. So originally in mitosis, we said that the sister chromatids, the chromosomes are going to line up single file. We had the polar. In meiosis, they were all vertically on this polar axis that I drew, single filed, and they got ripped apart like that. And this is going to be a little bit different from meiosis. So meiosis, the first step. So right now, this is the first phase. There's gonna be 2 phases. So 2 phases of each thing here. So what's gonna happen is these are gonna line up like this, how I have it. This isn't single file. I hope we can see that. I think you can. Okay. Yeah. There we go. All right. So these are gonna line up like this, and the reason they do that is because meiosis, like we said, is for your sex cells or the gametes. And the reason you need gametes to have half the amount of chromosomes is because the male sperm and the female egg are going to meet, and they need to create a cell, a somatic cell that is 46. So what they're going to do is combine the 23, I put 26, they're going to combine the 23 from the male, 23 from the mom, they're going to come together, and it'll have 46. And that cell can do mitosis and divide until it makes a human. So right now, we're at a somatic cell, and we need to make a gamete. And it needs to be genetically varied, I don't know if that's a word. Needs to be genetically different from its parent cell. And the way it's going to do that is you have your genes here. So let's see. So they're going to line up like this, and this is going to be the law. You need to know this for the test. Law of independent assortment. And the law of segregation. Okay, so we have your 2 laws that we're going to talk about, and you have your basic setup here. So this setup is going to be for the law of independent assortment. And we're actually going to talk about that second, because first we're going to talk about the law of segregation. So the way that we're going to get these different gametes to be genetically different is we're going to have random switch ups, and you'll see here. So we have our chromosomes in here, and what's going to happen, I don't think you can see them. What's going to happen is you have a chromosome here with its sister chromatids, and it's going to see another chromosome, and they're going to link up here, get close, and this is where the genetic variation part of it comes from. So what's going to happen is these 2 are going to pair up, and through the process of synapsis, I'll write this out, synapsis, they're gonna start to overlay on each other, so we can draw them closer. So they're gonna pair up like that, and then they're going to, it's kind of hard to draw here, but you're gonna overlap, something like that. And so you have this overlap here. And so right now synapsis is occurring because they're randomly switching the alleles. And this area here is called the chiasma. This is where they overlap. Chiasma. This area is called the chiasma, and you can see that forming. And through the process of synapsis, they're going to switch alleles, and they're at the same gene locus. So let's say you had it right here and right here, and they're gonna switch, or they might not. They might switch, and that's gonna happen. And so when they're done, you're going to have the same 2 chromosomes here, but one of them might have switched an allele, and so where this area once was might be over here now. So you have the red here, and instead of this being red now, it might be green with green eyes, or a new trait that it acquired from the other chromosome. I can actually demonstrate that with my pens here. So imagine this is one chromosome, this is another. You're gonna get together, you're gonna pair up, and you have your the same gene locus. So this is one allele, this is another allele, same trait, and they're at the same location. They're gonna overlap, and they will switch alleles. So now you have this chromosome, but it has this trait from here, and now this one has this trait from here. Okay, and so now they're gonna, that's synapsis also. That's the process of synapsis. Now you have genetically different chromosomes, and this is completely random, and that is the law of segregation. So now, once these are done doing that, that's when they're gonna line up like I had before. Switch the marker colors back here, confusing. And they're gonna line up, not single file like mitosis. They're gonna be a little bit different, like this. And this is the law of segregation. So you have your chromosomes with their different, it's really bad chromosomes, sorry. With their new genetic variations at them, and they're gonna line up like this, and this is the law of segregation here. So basically what's gonna happen is, like mitosis, they're gonna line up, and so whatever 2 are on this side are gonna become a gamete, and whatever 2 are on this side, they're gonna become a gamete. And so this is completely, it's pretty random. The exception is if there's 2 of the same, like this is Y, and this was big Y, you can't have them on the same side. But other than that, it's completely random. So let's say, we can actually label these. So we'll say this is the A, and we'll just call this one B here, and you have little b and A. Okay. So, or sorry, I did that wrong. Let's use different things here. Alright, so I went ahead and drew this out. So let's say this is A, this is little a, this is big B, this is little b. And so what's going to happen is these are going to kind of be randomly, and that's because of the law. So the law is going to determine randomly where these are going to go, and this gives it more genetic variation. So they switched their genes up, their alleles, and that gave them some genetic variation, and now they're going to give you more, even more for the gametes. So it could be A, B, and then A and B, sorry, big A, big B, little B for this gamete, and then that's how it'll be that gamete. So it could be switch, so it could be little a on this side and still be big B on this side. So there's all types of ways this could go. So you could have, let's say, you have gamete 1 over here and gamete 2. So 1 could be A, big A, big B, and then it could be little a, little b, or it could be big A, little b, or and if that was big A, little b, then this would be little a, big B. And you see all the different types. So you could have this one or that one, or keep switching it up, and it'd be multiple variations, and you get more genetic variation that way. And so that is meiosis, and eventually this is going to complete its process, and after all that happens through the law of segregation and the law of independent assortment, you're going to end up with 4 gametes. So you started with your 1 somatic cell, and then you went to 2, and then these went to 4, continuing the process. You don't need to know this, though. And now you have 4 gametes. So these are all gametes down here. Alright, and so the way we define these is through haploid and diploid. So we start with the somatic cell, which is our diploid, which has all the genetic information. And then we're going to end up going through with our haploid. So gametes are haploids, and the somatic cells are diploids. Basically saying that we went from 46 to 23 chromosomes. Alright,